Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, in the previous video, we discussed about DNA polymerase 1, 3 and DNA helicase. Today we shall discuss about the remaining enzymes that are also involved in the process of DNA replication in prokaryotes, for example in E. coli. First of all, we shall discuss about prokaryotic primase. Primase is the enzyme that is responsible for the synthesis of a short oligoRNA that serves as primer for the enzyme DNA polymerase. Primase play essential role in the initiation of replication at the origin, in the synthesis of Okazaki fragments, and also in the restart of a stalled replication fork. Prokaryotic primase consists of three domains. First is amino terminal domain. Amino terminal domain, it possesses a zinc carbon motif. This domain binds template DNA. The second domain is middle DNA polymerase domain, whose function is to polymerize. The third domain is amino terminal domain. The amino terminal domain either interact with a helicase or is itself an helicase. Based on structure and sequence alignment, primases are classified as DNA G protein. So this is about prokaryotic primase. Now we shall discuss about DNA ligase. DNA ligase is another enzyme that facilitates the joining of DNA strand by catalyzing the formation of phosphodiester bond. DNA ligase can repair single-stranded break and some of them can repair double-stranded break. DNA ligase play essential role in DNA replication and in DNA repair. For joining DNA strands, the enzyme DNA ligase obtains energy from the hydrolysis of nicotinamide adenine dinucleotide. So by getting energy from NAD, the enzyme DNA ligase catalyzes the formation of phosphodiester bond. The DNA ligase that is present in E. coli cannot ligate blunt-ended DNA fragments. It can ligate blunt-ended DNA, but only when there is molecular crowding. There is another ligase known as T4 ligase. T4 ligase is the enzyme that is present in the T4 bacteriophage. This T4 ligase can efficiently ligate blunt-ended DNA fragment. So friends, DNA ligase is necessary during the process of DNA replication in sealing the nicks that is very often created during the process of DNA replication. Another enzyme is single-stranded binding protein, SSBs. These are the proteins that bind single-stranded DNA, stabilize them and prevent their reannealing. These proteins are present in eukaryotes, bacteria and also in different types of viruses. Single-stranded binding proteins are essential in replication, recombination and repair. These proteins bind the single-stranded DNA and prevent their reannealing during the process of replication. In addition to this, single-stranded binding protein bind and modulate the function of other proteins. In E. coli, an active single-stranded binding protein consists of 4 identical 19 kilodalton subunits. Next we shall discuss about topoisomerase. Topoisomerase is an enzyme that catalyzes changes in the topological state of DNA. This enzyme catalyzes interconversion between relaxed DNA and supercoiled DNA. The main function of topoisomerase during the process of replication is to relieve the topological stress that develop due to the unwinding of DNA double helix by the enzyme DNA helicase. There are two types of topoisomerase, type 1 topoisomerase and type 2 topoisomerase. Type 1 topoisomerase bind and cut either one of the DNA strand, whereas type 2 topoisomerase bind and cut both the strands of a double-stranded DNA molecule. The main function of DNA topoisomerase is to relieve the topological stress through negative supercoiling. Friends, please share and like this video, and also please subscribe my YouTube channel Sam Online Class. Thank you very much. Bye bye. Good night.